based here, de novo prediction of subset localization, homology based prediction. Anybody volunteers? Oh, everybody's writing. No? Predict by homology, what does it mean to you? If I ask you what is homology based prediction of subset localization, what is it? That is a valid question, though. Really no idea? So, again, the idea of homology-based inference of subset localization, or of actually anything, homology-based inference of X, and the X be localization, homology-based inference means that you have a protein K with some known feature X, in this particular case subset localization, experimentally known, okay? And you have a protein Q that does not have known subset localization, for which you actually want to infer subset localization. So you simply look at the sequence similarity between Q and K. And if that is above some threshold, higher than some threshold theta to be worked out, then you say Q, my query, has the same subset localization as the known, experimentally known protein. Okay? That is by homology inference. What is de novo?
Well, in some cases you won't have any reference to get annotation from, so you have to predict de novo.
And how would you do that? Somebody else. How can you predict? In this particular case, it's subset localization. How could you predict it? So again, we have gone through the motifs, and I will get back to the motifs in a minute. But if you had no motifs, how could you do it? How can you develop a prediction method? By machine learning, right? Now, machine learning, any type of machine learning, what will it use? So it will have a data set. Say we will have a bunch of k's, so small k element of a bunch of k's here, a large data set of k's, proteins of known subset localization, and you will extract some common feature. Now, in this particular case, you will still actually, in some sense, maybe not by the same measure, but you will still look up the sequence similarity between your query and now the small k here, so the data set. If you found just this most similar one in terms of sequence in your data set, then that's homology-based inference. If you trained a machine learning device to somehow pick up an overall feature, that's machine learning. In the machine learning answer, typically, you will pick some signal that is not explicitly telling you it's this particular experimental information why I infer. But that typically is what people refer to as the black box. It's somehow hidden. What exactly makes that decision? And it could in fact be true that the information is contained in 5 different proteins that have different subset localization. It could be that part of it is clearly the same, this is not nuclear. And another one could be, this may be extracellular, this is not cytoplasmic. In 3 states you have settled it, although none of them in your database that made you make that decision was explicitly a good evidence for secretion or for extracellular localization, right? Now, again, the biological mechanism in the background is the idea of the signals that goes back to Günther Blobel. And again, the signals are not known for most proteins. So proteins are shuttled through the cell, eukaryotic cell, through zip code kind of motifs. We actually looked at them, signal peptides exist, we've looked at nuclear localization signals, and I've told you that this gets some way, but it does not get the entire way. In fact, not only does it not get the entire way, for most proteins we don't know the signals. Okay? Now what could it mean? It could mean that lots of these signals still have to be discovered. Or that there are other types of signals. It's a little bit this story here about sequence consecutive versus conformational. So maybe most signals are of the conformational type. That is in fact something that we do not know today. What we do know is we have very, very few examples that are clearly conformational. So somehow that's not really building trust. So most signals that we know, zip code-like motifs, are of this type. So if I assume now that most of those I don't know because most of those are conformational, it may be true, but odds are not really, our current experience does not really support this. What else could it be? There could be other mechanism. Can you think about another mechanism? So again, clearly some signals remain to be discovered. Clearly there will be some conformational signals. It's part of that, part of some other motif kind of story. And clearly there will be something else. Possibly there will be something else. Can you think about some other mechanism in which you could sort of indirectly use a zip code without directly using it?
Some chemical gradient?
Yes, that you're going too far. Yes, yes, you're right. But much simpler. Yeah?
Vesicular transport directionality is provided by codes, for example.
Yes, Jade, but this is still a zip code, right? This would still be in whatever we have, or to be discovered.
The signal is not in the sequence, but on the vesicle. So if you looked at the protein only, you wouldn't find it.
But it's an interaction between the two, right? Because it does not transport any protein. So there's the signal on the protein. It does not pick up any protein that comes close to it. It may not be a specific signal, like a zip code, you look at it and you know where it goes. So in some sense, I'm not entirely sure how to call that, it was like an active signal, right? And that, point taken. So there will be other motifs, and what you say in your point here, and it actually leads over to what I mean here, but this is neither conformational nor sequential, this is sort of an interaction type of motif. True, point taken. And think about that word and go one step further. That could be like this, right? You could have, this is my carrier protein, my shuttle protein. I have my nuclear localization signal, whatever signal you have. And you have some protein that binds to the one that has the signal. Okay? So by the green one binds to the blue. The blue one has the zip code. By the zip code, this one is shuttled, and the red one is shuttled with it simply because it binds. Okay? So in order to see how much that plays a role, what we need to do is find all the secondary cargo, so all the green ones, in this example that I had before.
Finally, it's always a motive, even if you use a shuttle protein.
So again,
protein has a motive.
Are you asking me or are you telling me?
Asking.
So again, I will come back to that later, but Blobel's discovery that gave him the Nobel Prize in 1999 was that there are these zip codes. You talk about the 2013 Nobel Prize, so the higher level of complexity, but there essentially are these zip codes that make proteins transported. There's a lot of experimental work that shows how these zip codes do exactly this transport, this regulation. Now the experimental work is providing a wealth of data that shows us how zip codes, direct or indirect, help to transport proteins through the cell. For most of these proteins that we know today, these zip codes are not known. Now what does it mean? It could be that there is something else out there, right? It could be that the many zip codes remain to be discovered. It's not easy to discover them. And that is what I'm trying to get at. Now the simplest mechanism that could be there to do this really is the cargo mechanism. Again, so we could have different types of motifs, we could have many motifs to be discovered, and we could have this situation, right? How could we estimate, how could we find all the secondary cargo? Or let's,
We would need to know all protein interactions, whether they are transient or permanent, and that's difficult.
So we don't know that. This is absolutely clear. My next question then is, and that is a question that is important to me, Because there's a question mark at the end, and you're highly invited to give me an answer, but I do not know the answer. Not only do I not know the answer, I have no clue how to address this. But this is something I'd like to sit down with somebody and think through. Maybe there is a way to address this question at least. Okay, we do not know all the proteins that interact. We know, in fact, we do not even quite know what fraction we currently know in terms of proteins that interact. So, but still, with all the data that we have about protein interactions and about motifs that we know, could we possibly come to an estimate of, say, a limit, an upper limit or a lower limit? Could we say, okay, it looks to me that, well, for 1/3 of the proteins we know the signals, and for another third we could explain it through secondary cargo. And that is something that I'd like to have as sort of a, well, this could be a master thesis because there is some method development underneath. I just want to phrase that question. It's an open thing that bugs me since a while. You want to comment on that, Jane?
Yeah, well, like there is an upper boundary. If you look at the compartment, we can see what proteins are contained there. We subtract those who already have a signal sequence,
yes,
because that will be secondary cargo.
Yes.
And we subtract those to be known, like to be produced in that compartment, and the rest would be the upper boundary then.
Yeah, well, okay, yes. So we know subset localization, I can't remember what the number is for human, for roughly 1/5 of all human proteins, and for, I cannot remember either that number, for maybe 16% or something less than 1/5, we know signals. So in some sense you have the upper boundary 80%. Can we get closer? Right? That's my question. And to some extent, so one way of approaching it would be the incomplete set of protein interactions. But there the tricky bit is to, in fact, estimate what the random expectation from an incomplete set would be. I believe this we could go to the next level. In the next level we would have to consider predictions. And now the other challenging bit is that proteins are travelers. So I'm talking about predicting subset localization as if a protein always stayed in the same compartment. They don't. They're all born on the ribosomes. So everybody's a wanderer, except for the few proteins that remain in the ribosomes, right? But when we talk about subset localization here, we always talk about the most dominant one. And that again is something that there's an important reality behind that. Anyway, let's put that to the side for the time being and talk about methods in which we can really predict for whatever is left based on some other feature. So again, let's just begin with the simplest view where we have three classes, nuclear, cytoplasmic, and extracellular. All we want to do is, without any motif, sort proteins into these three classes. How would you do that? Yeah, okay, there are prediction methods out there. But assume you were the first.
What he said, like if a protein interacts with another protein.
Okay. Let's go wind back time, right? So 2015, we do not have all proteins that interact. But this, what I'm going to show you, was the 80s, 90s, the first idea. So we clearly did not have all proteins then, okay? What's the simplest way in which you could possibly think to try to predict subset localization? What's the, how, I mean, yes, you would.
Text mining, when you have some papers.
Yes. So you have a data set. I'll give you a data set. Let's do that differently. Let me set up the experiment for de novo prediction in the way where I give you say 3,000 proteins, 1,000 for each class that are nuclear, cytoplasmic, and extracellular, 1,000 each. And let's say there is some sequence redundancy reduction done. So this is a relatively non-redundant data set. Next, how would you set up a machine learning device that predicts subset localization? So text mining in that sense, this is already, the text mining got me to that level. Okay? And again, 20 years ago, clearly the numbers were different. But let's assume this were your data set. How would you develop a method? What would you look at? What's the input to that method? Simple. What's the simplest thing you can think about?
A sequence.
How?
The word of sequences.
No, we have sequences. So sorry. Okay, I communicated incorrectly here. So this really means you have 3,000 sequences. But how do you use the sequence?
With like known localizations.
You have 3,000 known localizations. You have a data set with 3,000 properties for each one of those. You have the sequence. You have the known subset localization, 1,000 in each, just for the sake of the argument.
And I construct a classifier with some features like sequence similarity. Or
What do you mean by sequence similarity? You're going for homology based. No, it's not homology.
Or if it's not like that, use.
Tell me how you use homology. So again, it's not the type of thing where we say sequence similarity between the query and a particular protein of known, where k is one of these 3,000, is higher than a threshold. This situation, by construction of the data set, does not apply. Yeah?
Try to find some text fragments which appear only in the nuclear data set, for example. When you use text mining to derive the data set, you can try to derive some...
you mean sequence motifs?
words.
Is that what you mean? The text fragments? So yeah, I don't get...
about the paper.
You don't get paper.
Paper only.
So you... Come on, we're back to... The sequence contains the information. So you want to predict from sequence.
Okay, so you could look somewhere stream upward from your protein. You
Yes.
already mapped the protein to the genome.
Yes.
And then you can compare these regions and try to find similarities between all the stream upward regions of nuclear part, for example.
But that, again, so is a little bit, let me take you, this is a little bit late to make that point. This would be an alternative mechanism that you have in the back of your head, an alternative mechanism for sorting proteins. So implicitly what you're saying is that coming off the ribosome, you somehow, in the regulatory region upstream, you built in a mechanism by which, upon folding, you put a zip code before, whatever the mechanism is. But that is what you would have to postulate to exist in order to make sense out of what you say, right? Your biological intuition would only make sense if something like that existed. Now, I'm not saying that it doesn't. But at least we do not have evidence for that at the moment. We do have evidence for zip codes inside of the protein sequence. We do have evidence for sort of active zip codes, bless you. Back again. No text mining, no other annotation, no friend who will tell you, no existing method. You get the sequence, you get the classification. What feature are you going to look at that classifies your proteins into subset localization?
Check the fold and the physical or chemical properties of the surfaces. For example, if they are charged and the compartment has a certain pH.
Yes.
Like that.
Yes, Jade, all that stuff like that is already right. This one, scop folds, didn't work. Surface residues, let's delay that for a moment. We could do that. And pH is the real reason. But let's just be a value simpler before you check surface composition. What's the simplest thing you can think about for a bunch of sequences?
Amino acid distribution.
Come on, this is really the simplest, isn't it? So now you could imagine, since proteins consist of domains, in this particular case what I want to indicate here is a 3-domain protein, you have the choice. If you knew the domains, you could ask yourself whether you would actually sort of look at the amino acid composition of each domain. You could imagine that you run a window. The window problem is that somehow the signal can be anywhere in the sequence. Amino acid composition may be the dumbest thing to do, but certainly it's the simplest thing to do, right? And you could try to see whether, and the first thing to check really would be to see whether there's a correlation of amino acid composition and solvent accessibility at all. And that, in fact, is the work of Kenta Nakai and Kanhisha. Localization. In the first instance here, really, they just looked at amino acid composition, and then they began looking at more complex rules, such as N-terminal signals and the like, and membrane heases and all kinds, and that sort of evolved into a method that is one of the most frequently used methods to predict subcell localization, ultimately by Paul Horton, p-sort. And that is a combination of the simplest idea, really, amino acid composition, and the rules that exist. Now, again, do you believe that claim that amino acid composition is making the difference? What I show, and I'm apologizing for the quality, is right out of a paper from Miguel Andrade. First eigenvector, second eigenvector, amino acid composition. We're looking at 20 values, and we're looking at the combination of these 20 amino acids that makes the highest distinction in 3 different classes. The 3 different classes are blue, extracellular, red, cytoplasmic, and green, nuclear. And you see there is some separation. Again, it's difficult to see here. There clearly is green here. There's green in between 2 red clouds, so to speak. But there's also green here, and there's, for instance, no red and no blue here. So I believe this clearly establishes that amino acid composition distinguishes between the classes. Now here what I do is I look at the residues inside. The core residues, the residues that do not in fact touch the surface. And you may see, even at that resolution of the plot, that the classification is much worse here than here. So when you subtract one from the other, you see the surface residues. And in the surface residues, again, the plot is not very good, but you do see that there are green, red and blue regions, and that these are more apart, appear to be more apart than this. In fact, the classification is way better for the surface, which is exactly what you said, Jade. This is the difference in pH. Surface properties differ because the different subcell localizations have different pH. And so you see that imprinted upon the surface. This is a larger data set, slightly better view, and in this particular large data set, what we saw, for instance, in the middle of the red here, we had some extracellular proteins. Miguel found out that they are glycosylated. What's the significance of that? So you have to adopt with your surface composition to a different environment, so a particular environment. You put a sugar on top of it. That means you change your solvent accessibility. You change the surface. You change the pH properties of your surface. One sugar, a couple of sugar molecules completely turn you upside down in some sense, and you can suddenly, as a protein with a nice extracellular surface, so to speak, live in a cytoplasmic space by just putting a sugar. Perfect mechanism. And it's an interesting way to see how glycosylation is used to, in fact, make proteins fit to different compartments. Overall, there is a clear difference in terms of the preference of the surface residues for different compartments. And so let's just zip through this. So this means that in order to predict subcell localization, if you want to use it, you need to first predict solvent accessibility. Now, there are 2 different ways in which you can do that. So I thought that I give a brief run through the neural network and the hidden Markov model. It seems to be not the case. SVM, neural network, and the math underneath. Have we talked about it at all? Yes, in this context, in the functional class, right? So somebody, yeah. So what's the major difference between the 2? The math underneath is somehow similar. So let's first talk about the number of points, or the number of data points that you have. If you have fewer examples, why is one better than the other, and which one is better? If you have have more noisy data. Why is one better than the other? Which one is it? Okay, let's begin with, so essentially what the SVM does, it finds the points that are closest to each other, yet classified into different classes. Red, green, right? And tries to zoom into classifying those, arguing that these points down up here are distinguished anyway. Okay? Now this means, what does it mean in terms of number of points or number of samples you need?
When you have not enough points, you won't find a good hyperplane. And if you have
That is true for both of them. Let's talk about number of points first. Noisy will be the next question. Let's just look at number of points. The neural network finds a classification that optimally distinguishes all points. This distinguishes a subset of the points. So, immediately you see it. This can live with fewer points. On the onset, right from the start, from the get-go, it just throws away a lot of the points. The neural network doesn't do that. Okay, now about the noise. What does it mean in terms of noise? Assume you had noisy data. Which of the two can handle that better? And I pick these two extremes because they are very successful in the field, but also because in fact they just illustrate something very, very nicely, because both of them are really based on the same math in some sense. Both blow up the space in high dimensionality space and then find hyperplanes in the space. And they do the math underneath using essentially the same matrix conversions. It's like an eigenvector composition. Somehow the math underneath is very, very similar. That's why I picked these two. Yeah?
When you use all the points in the neural network, it's probably not so bad when you have noisy data.
Right.
Because you use all the points, and then you maybe average.
Sounds good. Sounds good. But there's something more essential. It's true what you say. The direction is also right. But the SVM is bad for incorrect data, and that is the major decision. So the direction is the same, right? Either the neural network is good or the other one is bad. But the major thing here really is the SVM is bad. Why? You find the points that are closest to each other and have different labels. That label is wrong. If this was a green one, right? Essentially what you're trying to do is you reduce the number of points and you rely very much on the ones that are most similar, that they in fact differ. Now if they exactly have the wrong label, then you keep trying to find a plane through the few points that are absolutely incorrectly labeled. That's for noisy data, or not only noisy. This could also be true that the way you decide what is green and what is red is based on something that is not quite physical. So what I mean, for instance, when you want to predict the context between two residues, so then you may ask yourself, what do you call a contact? How close do atoms have to be so that you call it a contact? Now, you may end up with a distribution on the C alpha residues, this around 8 angstrom. That happens to be the distribution you observe between C alpha and protein structures. Now, you may now decide, okay, my threshold is 8 angstrom. If you did that, then you immediately see that in some sense between 8.1 and 7.9 in terms of chemistry is almost the same thing. But then you would put this into green and red. You would call them by construction different things, although chemically they are very, very similar to each other. So putting it, not only that, putting the threshold between green and red into the peak of the distribution, you would increase the number of cases where falling on that side or that side is artificial, or is just random. Because if you were to put it here or here, the difference between 0.1 or whatever the difference is much less influencing your classification than if you put it into the peak. You all with me on that?
But if you use a probabilistic SVM where you assign probabilities for each class, it wouldn't be that bad.
be careful. The label is still black or white. The label is still green or red. You will not put a probability. Yes, so you allow the margin. Essentially what you have in your head is you make a larger margin. It's true. You train the SVM, the probabilistic SVM, in a way in which you say, okay, there's a bunch of points where I in fact allow this, but my point will remain if you did start it this way. Then essentially the most of the data is in the realm of what you call uncertain. So you make the margin very large. However, if you made the point, for instance, here, the cut here, you would still have the same sort of probabilistic network, but now most of the data would be used in either or the other direction. Would be actually used to train most of the important data, right? So whether it is black and white or gray level, ultimately, so how big your margin is, doesn't really matter to make that a bad idea. Back again. So having wrong data, SVMs work better with small data sets, and they work worse with incorrectly annotated data sets or very noisy data. Now, here's another question. Say we solved our problem with an SVM. And we had, in the neural network, for instance, we have 3 output states. In a neural network, this is easy to do. You have 3 output units. How can you binary classify? And I'm going to show you visually just neural networks because of historical reasons. But assume you have binary SVMs or binary decision systems or machine learners that have a binary classification. How can you create 3 classes?
You construct 3 one versus rest classifiers?
Yeah. So you say extracellular versus other, nuclear versus other, cytoplasmic versus other, and then you can put that in a different way. So you could put, say, sequence in, solvent accessibility in, and sequence and secondary structure in. You could merge all of that, so to have sort of a layered system. Could be a neural network in this particular case. It could be an SVM. Same thing, just for easy reasonability. I turn this upside down now. So again, they are all binary networks here. And now what I do here is I have 20 input units, as a composition, 20 input units, so surface composition. And here I have sort of a mix between sequence and secondary structure. Now if you knew that, as a composition, solvent accessibility and secondary structure are important, you could do it the way I do it, right? How else could you do it? How else could you use those information? Tell me when it's getting too cold. I try to optimize the oxygen signal. So I have 3 different machine learning devices trained on 3 different inputs. Because I know that as a composition is important, so surface composition is important, and somehow secondary structure plays a role. Could I have done it in a different way?
You could merge all the vectors and use it as one input.
How can you do that?
Just create more input nodes and concatenate the input vectors in some way.
So this is one solution. In this case, we have, let's just begin with the first 2 here. 20 plus 20, in your way, would be 40, right? Let's just look at secondary structure. That could be, say we talk helix strand other. How many would that be?
Different combinations. 4 different possible combinations of helix strand and beta sheet.
You said 4?
No. What was the question about this?
So, say I know secondary structure helps, and say my resolution of secondary structure is in 3 states: helix, strand, other. Those are my 3 secondary structure states. How would I use them as input? How many input units would that give me in your concatenated model?
Depends on the length of the protein and the number of...
Wait a minute. So far we looked at amino acid composition, surface composition. That does not depend on the length of the protein. Oh, I see what you're saying. Actually, your answer would create a problem, right? So in your answer where I simply count the number of helices, is that implicitly what you meant by your point?
Yeah, that would solve the problem. Or
no, that would create the problem. In my view, that would create the problem.
Yeah, but then you can normalize.
How?
Maybe just divide by the protein length.
Oh, so then you essentially look at amino acid secondary structure composition. What fraction of residues are in either of those three? Again, that is the way to do it. My question to you, how many input units do I have? Let's just look at what I call here the last neural network. In the last neural network on my slide, and again, any machine learning device, historically we began with neural networks. That's why they are there, because these slides are still from those days. In that one, I have sequence equals 20 amino acids, and secondary structure equals 3 states. How many input units are those? So there's one way, 60 or 23, right? Could be, you could try both. Why did you say 60, not 23? What's the advantage? Or what could be the advantage?
Maybe different amino acids occur especially often in helices, for example. And that you can take into account when you use 60 instead of 23.
That's one way of putting it. Another way of putting it is by creating 23 and 60. You create sort of two spaces of the input space with very different information content. So essentially the last 3 units would always be 1 or 0. And the first 20 units would also always be 1 or 0. The neural network itself sees individual units. And it will be complicated to sort of train, first of all, that there is a differential between the last 3 and the first 20. So you would put some burden onto the system that may be easier if you start with the 60, they treat it equally. And that is an advantage. And it turns out that the disadvantage of blowing up the input space in an environment of few samples was less important than the gain of sort of putting them orthogonal. In fact, that's exactly the way we did it. Now, using this is amino acid composition, this is surface composition, each one of them is 20. In the same logic, this could be 400. That did not work. Why? Why do you suspect?
Doesn't the quality of the neural network you train depends on the prior, and if you put like the buried ones in, it like reduces the quality of your result?
Be careful. With that same argument, this wouldn't bring anything, right? Essentially what you're arguing, if your argument were right, then this would not help at all. It does help. My question is, why in this, what we in fact did not consider is to do what exactly you proposed, the concatenation of 20 plus 20. We did not try that. Oh, I'm sorry, I do not remember whether we tried that. I don't have evidence. But we did try the 20 times 20. And why do you believe this did not work as well? Again, it cannot be your argument, Jade, because this one did work. I didn't tell you that. Could have been true what you said, but no, it's not true. So what else could you suspect? It's just too big. The space, we created a sequence space that was, in some sense, you may say this is related to what you call priors, but we just did not have enough density of the information to really support the data for the 400 units. As simple as that. While in terms of the blow up from 23 to 60. It's a factor of 3. But there it was still supported by information. Plus, by making it more orthogonal, the information was easier to use, obviously. So there was an advantage for solvent accessibility versus amino acid composition. It was not. Okay. And then you again get back to this issue of the 3 binary classifiers. Now, how would you, in fact, get from 3 binary classifications into 3 classes? So essentially, I'm sorry, maybe I was not clear. You have 3 different machine learning devices. One says extracellular and other, one nuclear other, one cytoplasmic other. How do you get the final prediction? So you run 3 machine learning devices, they get 3 results. How do you decide which of the 3 it is?
In the optimal case, only one predicts, not other, and in this case you can say it predicts nuclear and the other 2 predict other, so probably it's nuclear.
In the non-optimal case?
You have to flip a coin.
Oh, really? Yeah, that. Okay, that. You give me 2 examples, 2 extremes in some sense. Well, let's just give you an example. Does that give you an idea how to proceed? So the first thing I want to point out, okay, can somebody say something?
Yes, you can say that it's not the first one.
It is not extracellular. Yes, that's true. We have. Do we have to flip a coin?
Not really, because we have numbers.
Yes, and?
So we choose cytoplasmic.
We choose cytoplasmic, and the argument being?
Because the 0.7 is bigger than 0.6.
Yes, and I made a mistake again. The last time I translated this from the whiteboard. On the whiteboard it worked, but I made a mistake in writing it down. So what I believe what I wanted to induce in you first is many machine learning devices do not put out probabilities. You see this, they don't sum to 1, right? So going for the highest number, my example should have been chosen such that the highest number is not the right answer, but that you have to first normalize it. Okay, in this particular case, I messed up. Okay, but again, so yeah, the simplest way is in fact you go for the maximum probability answer for the strongest answer, and essentially this is sort of summed up here. Now another way of doing it. Next Tuesday, because we need 15 minutes for questions, or we go further? Another way of doing it, so you know there's the biological pathway, or the biological way of sorting, that has a priority. There's the secretary pathway here, there's the gated transport between cytosol and nucleus, and there's the separate sorting into mitochondria. And you could simply imagine that you put this into, that you put your prediction device onto such a hierarchy that mimics what we observe. So in that hierarchy then, we have let me just look at it, the advantage now. Say the first decision is, are we going into the secondary pathway or not? All right? And I train, this happens to be an SVM, so neural network or whatever, machine learning device, for this decision. And then I go further. Do you see the downsides and upsides of this solution? So this is, again, I'm not training one against everything and then I go for the max. I use a hierarchy. What is the beauty it buys me, and what's the problem it may buy me or get me into?
I see a problem with like the first SVM does a wrong prediction, then you can't get correct.
Exactly, you're trapped. Let's get back to that point at a later point. But yes, that is a downside. You don't see anything positive? Oh, come, don't be so negatively picky. Nobody sees an advantage of this scheme?
Yes, you have a result in every case. So you don't have
To flip a coin.
Yes, you don't have to flip a coin.
Well, in the other one you also had a result, according to what you said is the maximum number. There's only one max. And if yes, you could construct a case where the max is really is a tie, but this could also be a tie.
Maybe this saves time, like,
It absolutely saves time.
because you don't have to use all the proteins.
That, in fact, is an important aspect. But we will get back to that issue. You will lose that again. But it's true. But there's something else. Much simpler.
Include more information.
Yeah, how? If you know what you mean, then you gave the right answer.
Yeah.
How do you put in more information? That is true.
I'm not sure how you derived information about the
More
secretary.
specific. Let's call it more specific information. The point is that you complete these machine learning devices. They may in fact not even be SVMs. This may be an SVM. That may be a tree, decision tree. That may be a random forest. You can completely tailor. The beauty of this is you can tailor the particular prediction task at hand to the data set there. So this one, for instance, we looked at signal peptides. This could be a system that only looks at the N-terminus. First 100 residues or 70 residues. All right? This could look at the entire amino acid composition. This could look at surface composition. Whatever is relevant for that, for that example. That's the first advantage. The second advantage is that the further down you go, the more it becomes relevant that you look at data which is specific to that distinction. So this thing down here will never get nuclear proteins. Maybe distinguishing, when you have to distinguish between extracellular proteins and other vesicle transport, maybe this is a really tough problem. And then if you threw nuclear proteins into that mix, then it would just zoom into the nuclear proteins and say, well, these clearly are not our case, and this will dominate the prediction. Now we can no longer have this problem, because now we only get the two data sets that are somehow similar. They are both not nuclear. They're both not easy to set apart. Whatever it finds here is really relevant for the detailed distinction, right? That's the other advantage. So the further down you go, the more information that is specific to that distinction can you put in, the more you can adjust your system to that distinction. Now, initially, this in fact is very much what we did. We put a very, very different information into different SVMs here. Now, one issue, the bullet points here only say what I already said. One issue that we have to remember is getting tougher, because we're getting down to proteins that are more and more similar to each other by construction, right? So the tasks get more difficult. Let's just look at the performance. Tougher at the top, there's the highest level of performance, and then performance further down goes down. And let's just keep it there and move onward to some other methods. But I believe all of these other methods, let me put this into the remaining time and still allow a few questions by saying there are many other methods out there. So what I showed you is just one idea. And they use different pieces of information. To make you happy, Michael, some of these use text mining. It's simply because you said that 3 times today. I'm sorry. Even when I told you we have this sequence, you still wanted to go back to. So let's just go in. One of those, p-sort is what I said historically. So that's back to Minoru Kanaisha. And by the way, Minoru is also the father of KEGG, which he is a giant in the field. Kenta Nakai is another shaper of the field, and the third one that is looking at these rules is Paul Horton, who connected rules and simple approaches to machine learning. That's p-sort, one of the most often used, that also uses k-nearest neighbors. And now it's called Wolf p-sort. Then one of the other methods, the signal peptides methods we talked about, this here is exactly the idea. that combines, Sherlock is what it's called. It gets a high accuracy by combining machine learning of sequence information with PubMed abstracts. A very interesting way of doing that. And using motifs and using everything into machine learning. And there are different ways of doing that. And they have different tricks. And they have different, for instance, here for cello, the particular way in which it does the Kamer segments is interesting and unique. And I leave it here, and I'm going to talk next Tuesday, because Thursday is the Dies Academicus, so you're going to listen to talks down somewhere else, downhill. Downtown. And what I'm going to talk about is one other method that uses sequence in a slightly different way to predict subset localization. There's a lot to be learned from these methods, but I believe the major principles are established. The Kamer idea that is somehow phrased here, I will pick up on Tuesday and present another method. Some method, and let me just say that and show Mikhail's face. Mikhail is an expert on, he's in Queensland University, Brisbane. He clearly is a shaper in the field. He became an expert on Bayesian networks and statistical motif analysis. One tool that comes from his meme is one of the best motif finders out there. Or the math underneath meme is partially back to Mikhail Bodin. And he worked also on a prediction of sorting into subnuclear compartments. So I said 3 states. This now goes into nucleus, has a structure, and there's a very complicated, he did an outstanding piece of work on, in fact, sorting things into subnuclear compartments. I will just leave it here. And the reason is somehow I'm behind the schedule that I set for myself in this lecture. So I'm fast forwarding to get to next Tuesday, where we still do subset localization. Then you present your talks on Thursday. And I then want to get into protein interactions, which in fact will only be Thursday and a week from now, because next Tuesday, the Tuesday after the one, where the 15th of December, they will be data mining for health. Gosh, time. Wait a minute. Do I? Poof. I wanted to do much more than we have time for. So all of this is because I forgot that we actually have Christmas already. Essentially, wait a minute, this one we cannot do because you present. This one is subset localization, so there is no interaction. Okay.
We can just cancel our presentations.
Do you want to?
Sure.
Really?
Yeah. I don't know. We can present our presentation in the tutorial, so not in the lecture also.
No, that we cannot, because I have an alternative. I'm busy at that time, so I cannot be here. And I want to hear it.
You want to?
Yes.
That's the problem.
Let's just get the camera off.